To begin our discussion on the management of the RootSmart for optimal air pruning, I just wanted to reference our user guidelines document for some background information and for some context, as a significant portion of that guideline actually outlines RootSmart best practices in a way that really speaks to watering that's optimal for root pruning. So I've pulled the two most relevant points with regard to watering from that document, which I'm going to highlight here before we get into the details of the watering data collected off of the RootSmart apples. The first management strategy that's unique to RootSmart is cyclical watering, which just refers to less watering more frequently to minimize overwatering and allow for that characteristic dry out that's associated with air pruning in the RootSmart. The second management strategy that I wanted to highlight is low edge blocking, which just refers to blocking or configuring the trays by species with minimal edge to interior ratio. And what that does is allows for us to minimize edge dry out, both at the block level as well as at the tray level. These two management guidelines that are specific to RootSmart are particularly important to keep in mind as we evaluate the results of this apple pilot trial. But full user guidelines are available at the Greening the Canadian Landscape website. So the apple water monitoring trial included 5 EC5 sensors that were installed for 12 weeks between the 25th of February and the 19th of May to measure percent volumetric water content in 5 spatially distinct RootSmart cells, which you can see outlined in the diagram at the bottom left-hand side of the slide. The resulting data is presented here at the top of the slide as a time series with hourly percent volumetric water content measurements plotted as a function of date and annotated week of the trial, where week 1 is the week of the 25th of February and week 2 is the subsequent week, and so on. Also on that figure, you'll note 3 key thresholds that we're using to evaluate watering efficiency, specifically water holding capacity in blue, wilting point in red, both of which refer to the maximum and minimum water potential of the grow mix substrate, as well as the optimal saturation range in yellow, which is between 15 and 25% volumetric water content. All the values that I have displayed on these figures are estimated values according to our prior experience working with the substrate and working with the RootSmart tray, but more detailed data pertaining to the water holding capacity of grow mix can be obtained. And that's something that we're interested in doing moving forward using the HiProp2 and WP4C that we have and that we're always super enthusiastic about in our greening lab. So the data that's summarized here is an average of the measurements that were collected by the 5 EC5 sensors and constitutes what I think is representative of watering at the tray level as opposed to the individual cell level, which I'll cover off shortly. As you can see, watering was consistently within that optimal saturation range between 15 and 25% volumetric water content, and that there was minimal over and under watering over the course of the 12-week trial, where instances of overwatering and underwatering were not just minimal in their frequency, but also really minimal in their magnitude, where you can see that average overwatering and underwatering only just exceeds or just falls below that recommended range, and is more so just outside the optimal range as opposed to ever being particularly critical. Another important thing to note from this data is that we were able to successfully increase the frequency of watering over the course of the trial while maintaining that characteristic cyclical watering regime of drying out and rehydrating within that optimal saturation range, instead of resorting to having the Ellepots being super hydrated to avoid dry out as the water demand increased over the course of the trial. So overall, the primary takeaway from this average watering data is that watering was really very well done, and it followed that cyclical, high-frequency, low-volume watering regime that really underlies optimal root pruning in the RootSmart tray. So this data summarizes the percent volumetric water contents for each of the 5 EC5 sensors and constitutes an individual watering profile of the 5 spatially distinct cells. So whereas before we were looking at the average of the 5 cells, we're now looking at the individual cell data. One thing to note here is that watering is quite a bit more variable at the cell level, where you can see more frequent over and under watering events with greater associated magnitudes. So to get a little bit more into that detail here, I'll draw your attention to the yellow line along the top of the series, which corresponds to P4, which was an edge cell that was blocked at the center of the tray configuration. And what you can see is that this cell has a tendency for watering above the recommended range, likely due to its interior block position. So the fact that it's sort of located at the middle of that block. The pink line corresponds to P3, which was both slightly under and overwatered early on in the trial. And again, that variability is likely due to its edge position, this time at the edge of the tray, as well as at the edge of the block. The green line corresponds to P1, which had the tendency to be underwatered and at some points critically so. Going into this trial, this was the well that I expected to be the most vulnerable to dry out, given its edge position both at the tray and the block level, in addition to its proximity to the greenhouse door. And as we can see, it's consistently drier as compared to other wells, where you can follow that P1 series along the bottom of the data set over the course of the 12-week trial. Finally, the blue line corresponds to P5, which is also an edge cell that was blocked at the center of the tray configuration in a similar way to P4. However, P5 was consistently and often critically underwatered. And this discrepancy in watering might be capturing another really relevant factor that's contributing toward effective watering, which is the variability in how the hose or the boom is actually able to access the trays. And so in my mind, P5 may have just been in a bit of an awkward spot where there was difficulty maneuvering the hose or reaching over the bench to access and water that section of the trays. Now, this data that outlines the watering profiles of the 5 spatially distinct wells more so than the average watering data is really highly specific to this particular block configuration combined with this particular set of watering practices and these specific operators. And so my primary takeaway that I wanted to highlight here is the value of monitoring percent volumetric water content, whether it's done visually or with a sensor, not just with respect to time, where you're checking the percent volumetric water content at different time points to monitor the dry out, but also to monitor things spatially. So that would really allow for you to keep track of that spatial variation and perform more informed, targeted, and optimized watering, and that it really allows for the identification of these little watering anomalies that can potentially affect the overall watering success. So to wrap things up, I just wanted to quickly highlight how the findings derived in this current trial align with some previous work on watering in the RootSmart system, which was started way back before my time in 2016, when the team used the same EC5 sensors to monitor dryout across blocks of RootSmart trays at Vineland. Overall, what they found, and what you can see in the figure on the slide, is that the trays located at the interior of the block, indicated in both the diagram as well as the figure in blue, are so well insulated that they take a really, really long time to dry out, as you can see from that small negative slope. Now, as you move more progressively out from that interior, you'll note that the rates of dry out actually increase quite rapidly, with the mid-block trays in orange drying out relatively fast, and those edge trays in red even faster than that, again, indicated by that steep or high negative slope. These findings contributed toward the second management strategy that I outlined at the beginning of this presentation, namely low edge blocking, where the second configuration of trays, or configuration B on the slide, minimizes the number of edge trays as compared to configuration A, where both configurations have the exact same number of trays. The findings that were derived from this 2016 trial at Vineland were then carried over and used to inform a subsequent watering trial that was conducted in 2017, this time at the nursery. And so the nursery propagation of 4 species in the RootSmart trays. These were 4 tree species that were used in the field establishment trial that we currently have ongoing at Vineland on the home farm. Working in collaboration with the nursery, the team set up RootSmart trays in that B configuration from the previous slide to minimize the edge to interior ratio of the block trays. In the case of this trial, cyclical watering proved to be a bit of a challenge, where in trying to navigate a new watering regime for an entirely new tray that was populated with 4 different and unique species, the growers ended up watering so much to accommodate the edge dryout that the majority of trays ended up being consistently and critically overwatered, and overwatered much more than anything that we observed in the current apple RootSmart trial. And as a result of that overwatering, the interior trays were continually hypersaturated, while the exterior trays fluctuated between being hypersaturated and extremely, extremely dry. And in both of these circumstances, the absence of effective watering worked to inhibit the air pruning function of the tray, resulting in the occurrence of root level defects that the tray is specifically designed to deter. Example of which is the descending root that you can see on the slide, which is far more characteristic of a tray with a solid plastic wall than it is an open-sided tray like the RootSmart tray. And this brings us to the next section of the presentation, where we'll talk a little bit more about the importance of root morphology and how it can be influenced by propagation tray design.